Hello, I'm Neil Clark, and in this talk I'm going to talk about data normalization. I'll start by giving a definition of what I mean by normalization, and before giving you a quick demonstration of the need for normalization. Then I'm going to give you a kind of a general framework for data normalization before talking about some specific methods for microarray and RNA-seq data. Quantitative biological experiments are designed to measure quantities of biological significance, which means that we're typically in a situation where we attempt to interpret variation in the data in terms of the underlying biology. However, some of the variation in the data comes about not from underlying biological variation, but actually from non-biological sources. Some examples in the context of microarray experiments of this come from biases in the image intensity that come from overall spot intensity, spatial location on the array chip, and scanning artifacts. RNA-seq technology is often portrayed as a solution to many of these problems. However, even this technology has its own non-biological sources of variation. These could come about from various biases. Some examples of non-biological sources of variation in RNA-seq data are the sequencing depth, so how many reads are counted in a given lane, and gene-specific biases that are perhaps related to their length. And there are also biases that are related to the guanine cytosine content. Now, the aim of normalization is to remove as many of the non-biological sources of variation as possible, and this allows a comparison between biological samples. This is an extremely important step in the pipeline of the analysis of biological data, and all subsequent biological influence is dependent critically upon this step. If it is not done properly, you can get some very inaccurate or potentially misleading conclusions from the inference. So here, let's take a look at a more specific example of the need for normalization. I'll do this in the context of a two-color microarray experiment. Now, the kind of technical variation or non-biological variation that we're going to examine here is an intensity-dependent relationship of the fold change of the red and green channels in the microarray. We can demonstrate this by using what is called an MA plot. So here, for a given sample, for each probe, we plot the ratio of the red and green intensities against the product of the red and green intensities, and we do this on a log scale. So this is shown in the top right figure here. So what we see is that there is a curve with a defined shape in this plot. That means that there is some kind of systematic dependence of the fold change upon the overall intensity. Now, this kind of variation is non-biological. It's a variation, a dependence or a bias that comes of a technical origin. The normalization procedure attempts to find a transformation of the data that removes this variation. So effectively, we go in from the top figure to the bottom figure. So here in the bottom figure, you see that the MA plot is flat. So there is no dependence of the fold change upon the overall intensity. So the result here of normalization is that we have removed one of the non-biological sources of variation. So what we're hoping that what is left after this transformation of the data, the variation that is preserved, is a variation that is of a meaningful biological origin, upon which we can base further analysis.